Let's look at the physiology of fertilization. If we look at this slide, we have the time course of conception. Basically, here's the ovum being released into the fimbriated end, and fertilization usually occurs sometime in the first 24 hours after the little ova is ejected into this. So it's important that intercourse occur very shortly before or just after ovulation. We don't have a big window there. The reason is the sperm cannot live for more than a few days. So they need to get through the uterus, up into this tube, and get fertilized just either side of ejaculation. Well, not either side of ejaculation, but either side of this small window in which the ovum is moving down the tube. Finally, divisions start occurring here. You have first cleavage completed, and so that we have a two-celled conceptum here, then 4 cells and so on. And by the time we get down to the 4th day, we have something called the morula, which has hundreds of cells at this point. Then something called a blastocyst, which is a sphere of cells with a hollow center, occurring sometime around after the 5th day. And then toward the end of the first week, the blastocyst will implant into the side of the uterus. If the uterine wall is not perfectly developed to accept it, then a spontaneous abortion will occur, and this will present itself as a normal menstrual cycle. The patient may never know that they were pregnant. They'll start bleeding a week or so after they've ovulated, and they'll think they just had an early period. That's called a missed abortion. Now, when we move on to what's happening in the implanted ball of cells, we get something that looks like this. The hormones now are rising at a very steep rate, and we have something called prostaglandins in the semen that can stimulate uterine contraction and get this ball right to where it wants to be. And we have the cells here. This is the uterine cavity side, and then we have the lining of the uterus, and these are both replicating at a very fast rate. We have now the ball shown here with its hollow single layer of cells on the outside, and then an inner cell mass here that is beginning to differentiate. And finally, at about 7 days after fertilization, we're definitely getting 2 layers of cells here. And notice what's happening to the endometrium. We're starting to get cells here that are differentiating as well. We're bringing a huge blood supply to this area, and we have what is a benign tumor, the implanted blastocyst, eating its way into the wall. This is the only invasion of one tissue into another that occurs in benign, quote, disease. It's a benign process, but it mimics the invasion of a cancer. There's something called capacitation, in which a group of enzymes secreted by all the sperm, as you remember, have gotten in and weakened the wall of the ovary. That's what's allowed them to get in. And then there's an enormous number of changes mediated by a lot of different enzymes and hormones that nurture everything that's going on in both the mother's side and on the child's side, at this point just a blastocyst. The implantation continues until you have several membranes here, and we have a huge circulatory system that's parallel. And it's like many of our other circulatory systems. There should not be the mixing of the mother and the baby's blood at this point. The placenta, which develops on the maternal side here, which is going to be the future placenta, and the umbilical cord that links the baby are going to run their cells past each other, very close, again, 1 capillary apart, so the mother will exchange all her gases into the baby's blood, take out the baby's carbon dioxide, give the baby oxygen. The baby's heart and lungs The heart will be pumping, but the lungs will not be functioning. We're going to bypass the baby's lungs because there's no air in it. The blood is not yet mixing, and we don't want it to mix because there may be blood type differences from the mother to the baby because of the father's contribution that might be incompatible. We get sugar exchange, we get the exchange of nutrients and the exchange of wastes. That all goes across the two membranes, but not red cells and not the antibodies of the blood system. The mother can give the baby antibodies. The mother can give the baby antibodies in milk too. But this blood system is not going to mix because it may be incompatible. Toward the very end of pregnancy, the uterus starts undergoing a few contractions. The placenta starts to age, and few of these blood cells and blood vessels can break down, and there can be a little mixing. And that's why when we get blood type incompatibility, we can get some of the mother's blood into the baby's system, breaking down some of the baby's blood cells, which are seen as foreign blood cells in the system that's now leaking. And that's when we get into the very late problems of neonatal jaundice, which can occur from usually the Rh incompatibility in blood. Those don't occur usually till very late, and by then the baby can be delivered. And we will talk more about delivery and some of the problems of the genetic side in our next